तो यारो आज हम पढ़ने जा रहे हैं चैप्टर 1 सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट। और थोड़ा सा पहले इन महाशय के बारे में पढ़ लें। पंचानन महेश्वरी जी के बारे में भी पढ़ लेते हैं पहले थोड़ा सा, है ना। चलो स्टार्ट करते हैं। Born in November 1904 in Jaipur, Rajasthan, Panchanan Maheshwari rose to become one of the most distinguished botanists in not only of India but of entire world. He moved to Allahabad for higher education, where he obtained his DSc during his college days. He was inspired by Dr. W. Dugan, an American missionary teacher, to develop interest in botany and especially morphology. His teacher once expressed that if his student progress ahead of him, it will give him a great satisfaction. These words encouraged Panchanan to inquire what he could do for his teacher in return. He worked on embryological aspect and popularized the use of embryological character in taxonomy. He established the development of botany University of Delhi as an important center of research in embryology and tissue culture. He also emphasized the need of initiation of work on artificial culture of immature embryos. These days, tissue culture has become a landmark in science. His work on test tube fertilization and in vitro pollination won worldwide acclaim. He was honored with Fellowship of Royal Society of London (FRS), Indian National Science Academy and several other institutions of excellence. He encouraged journal education and made a significant contribution to school education by his leadership in bringing out the very first textbook of biology for higher secondary school published by ncert in 1964। चलो जी अब बढ़ते हैं अपने चैप्टर की ओर। सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट। आर वी नॉट लकी दैट प्लांट रिप्रोड्यूस सेक्सुअली? The myriads of lover that we enjoy gazing at, the scent, the perfume that we shone over, the rich color that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness, or flowering plants shown sexual reproduction. A look at the diversity of structures of inflorescence, flower and flower plot shows an amazing range of adaptations to ensure formation of and product of sexual reproduction, the fruit and the seed. In this chapter, let us understand the morphology, structure and the process of sexual reproduction in flowering plant angiosperm. Flower, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time memorial. Flowers are object of aesthetic, ornamental, social, religious, and culture value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. Let at least five flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the name of five more flowers that are used in social and cultural celebration in your family. Have you heard of horticulture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. In earlier classes, you have read the various parts of flower. Will have you called the part of a typical flower. Can you name the two parts of a flower in which the two most important units of sexual reproduction developed? Pre-fertilization structure and event. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed, which bear the floral buds, and then the flower. In the flower, the male and female reproductive structures, the endoecium and the gynoecium, differentiate and developed. Would you recollect that the androecium consists of a row of stamens representing the male reproductive organ and the gynoecium represent the female reproductive organ? Stamen, microsporangium and pollen grains shows the two parts of typical stamen, the long and slender stalk-like filament and the terminal general bilobe structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and the length of stamens are variable in flower of different species. If you were to collect a stamen each from 10 flower, each from different species, and arrange them on a slide, you would be able to appreciate the large variety in size seen in nature. Careful observation of each stamen under a dissecting microscope and make neat diagram would elucidate the range in shape and attachment of anther in different flower. A typical angiosperm anther is bilobed, with each lobe having two theca. They are dithecous, often a longitudinal groove run lengthwise separating the theca. Let us understand the various type of tissue and their organization in transverse section of an anther. The bilobe nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the cornel, in two each in lobe. Microsporangia developed further and become pollen grain, pollen sac. They extend longitudinally, although the length of an anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears nearly circular in outline. It is generally surrounded by a four walls layer: the epididymis, endothecium, middle layer, and the tapetum. The outer three layers perform the function of protection and help in dehiscence of anther to release pollen. The innermost layer is the tapetum. It nourishes the developing pollen grains. Cell of the tapetum possesses dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue, occupying the center of each microsporangium. Microsporogenesis. As the anther developed, the cell of sporogenous tissue undergo mitotic divisions to, to form microspore tetrad. What will be ploidy of the cells of the tetrad? As each cells of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen of microspore mother cells. The process of formation of microspore from a pollen mother cells (PMC) through meiosis is called microsporogenesis. The microspore as they are formed are arranged in a cluster of four cells, the microspore tetrad. As the anther mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspore or pollen grains are formed. that are released with the dehiscence of anther. Pollen grains. The pollen grains represent the male gametophyte. If you touch the open anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your finger. Sprinkling the grains on top on a drop of water, take on a glass slide and observe under a microscope. You will really You will really be amazed at the variety of architecture, size, shapes, color, design seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. It has a prominent two-layer wall. The hard outer layer is made up of a sporopollen, which is one of the most resistant organic material known. It can withstand high temperature and strong acid and alkali. No enzyme that degenerates sporopollen is so far known. Pollen grain enzyme has prominent aperture called germ pore, where sporopollen is absent. Pollen grains are well preserved as fossil because of the presence of sporopollen. The exine exhibit a fascinating array of patterns and designs. The inner wall of pollen grain is called intine. This is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserved and a large irregular shaped nucleus. The generative cell is smaller and floats in the cytoplasm of the vegetative cell. It is spindle shaped. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm pollen grain are shed at the two-celled stage. In the remaining species, the generative cell divide mitotically to give rise to the two male gamete pollen grain are shed three-celled stage. Pollen grains of main species cause severe allergy and bronchial afflictions. In some people, often lead to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminated with imported wheat has been ubiquitous in occurrence and cause pollen allergy. Pollen grains are rich in nutrition. It has become a fashion in recent years to use pollen tablets as food supplement. In Western country, a large number of pollen produced in the form of tablet and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. When once they are said, when once they are said, pollen grains have to land on stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viability is highly variable and to some extent depend on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some member of Rosaceae, Leguminosae, and Solanaceae, they maintain viability of month. You may have heard of stored semen, sperm, or many animals, including human, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen minus 96 degrees Celsius. Such a stored pollen can be used as pollen bank, similar to seed bank, in crop breeding program. The pistil, microsporangium, ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When they are more than one, the pistil may be fused together, syncarpous, or may be free apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulge part of the pistil is the ovary. Inside the ovary is the ovarian cavity locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placenta that you studied in class 11th. Arising from the placenta are the megasporo- megasporangia, commonly called ovule. The number of ovule in an ovary may be 1, wheat, paddy, mango, to many, papaya, watermelon, orchid. Megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fused with funicle in the region called hilum. Thus, hilum represents the junction between the ovule and funicle. Each ovule has one or two protective envelopes called integument. The integument encircle the nucleus except at the tip of where a small opening called micropyle is organized. Opposite to the micropylar end is the chalazal, representing the basal part of the ovule. Enclosed within the integument is a mass of cell called nucleus. Cells of the nucleus have abundant reserve food material. Located in the nucleus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cells is called megasporogenesis. Ovule generally differentiate a single megaspore mother cell (MMC) in the micropylar region. In the micropylar region of the nucleus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes mitotic division. What is the importance of MMC undergoing meiosis? Meiosis results in the production of 4 megaspore, female gametophyte. In a majority of flowering plant, one of the megaspore is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy of the cells of nucleus, MMC, the functional megaspore and female gametophyte? Let us study about the formation of the embryo sac in detail. The nucleus of functional megaspore divide mitotically to form two nuclei which move to the opposite pole. Formation: the two nucleated embryo sac. Two more sequential mitotic nuclear divisions result in the formation of four nucleated and the later enucleated stage of the embryo sac. It is the interest of to note that the mitotic division are strictly free nuclear. That is, nuclear division are not followed immediately by cell wall formation. After the 8 nucleated stage, cell wall are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac. 6 of The 8 nuclei are surrounded by the cell wall and organized into cell. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cell. Within the embryo sac, 3 cells are grouped together at the micropylar and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergid have spatial cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tube into the synergid. 3 cells are the chalazal and other the antipodals. The large central cell is mentioned earlier has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8 nucleated, is 7 celled. Pollination. In preceding section, you have learned that the male and female gamete in flowering plant are produced in the pollen grains and the embryo sac, respectively. As both type of gamete are non-motile, they have to be brought together to fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is stem pollination. Flowering plants have involved in amazing array of adaptation to achieve pollination. They may use of external agent to achieve pollination. Can you list the possible external agents? Kind of pollination. Depending on the source of pollen, pollination can be divided into 3 types. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to stigma of the same flower. In a normal flower which opens in exposes the anther and the stigma. Complete autogamy is rather rare. Autogamy is such flower required synchronine pollen release and stigma receptivity, and also the anther and the stigma should lie close to each other so that cell pollination can occur. Some plants such as Volia, common Pansy oxalis and common Nilla produce 2 types of flower. Chasugamous flower, which are similar to flower of other species with exposed anther and stigma, and celastogamous flower which do not open at all. In such flower, the anther and stigma lie close to each other. When anther dehiscence in the flower bud, pollen grains come in contact with the stigma to effect pollination. Thus, celastogamous flowers are invariably autogamous, as there is no chance of cross pollination. No chance of cross pollination landing on the stigma. Celastogamous flowers produce a short seed set even in the absence of pollinators. Do you think the celastogamous is advantageous or disadvantageous to the plant? Why? Geitonogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Although geitonogamy is functionally cross-pollination involving in pollination agent genetically it is similar to the autogamy since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grains from anther to the stigma of a different plant. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agent of pollination. Plant use two biotic wind and water and one biotic animal agent to achieve pollination. Majority of plant use biotic agent for pollination. Only a small portion of plant use abiotic agent. Pollen grains coming in contact with this stigma is the chance factor in both wind and water pollination. To compensate for the uncertainties and associated loss of pollen grains, the flower produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. Wind pollination also requires the pollen grains are the light and non-stuggy to the that they can be transported in wind currents. They often possess well-exposed stamen so that the pollen are easily dispersed into wind currents and large, often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The ears you see are nothing but the stigma and the style, which wave in the wind to trap pollen grains. Wind pollination is quite common in grass. Pollination by wind is quite rare in flowering plants as is limited about to 30 genera. Most monocotyledon as against this. You would recall that water is a regular mode of transport of the male gamete among the lower plant groups such as algae, bryophyte and pteridophyte. It is believed, particularly for some of bryophyte and pteridophyte, that their distribution is limited because of the need of water from transport of male gamete and fertilization. Some examples of water pollination plant are Valsneria and Hydella, which grown in fresh water and in several marine seagrass such as oyster. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth, water lily, the flowering emerges above the level of water and pollination by insect or wind as in most of the land plant. In Wilson area, the female flower reach the surface of water by the long stalk and the male flower of pollen grain and release it onto the surface of water. They are carried passively by water currents. Some of them eventually reach the female flower and the stigma in another group of water pollinated plants, such as seagrasses. Female plants remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long ribbon like and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by a miscellaneous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? Majority of flowering plants use a range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds. And bat are the common pollinating agents. Among the animal insects, particularly the bee, are the dominant biotic pollinating agents. Even larger animals such as some primates, lemur, arboreal, tree-dwelling rodents, or even reptiles, gecko lizard and garden lizard, have also been reported as pollinators in some species. Often flowers of animals pollinated plants are specially adapted to a particular species of animals. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small in numbers of flowers are clustered into a inflorescence to make the conspicuous. Animals are attracted to flowers by color and or fragrance. The flower pollinated by flies and beetles secrete fall odors to attract these animals. To sustain to sustain animals visit to sustain animal visit the flower have to provide reward to animals. Nectar and pollen grain are the usual floral rewards. For harvesting the rewards from the flower, the animal visitors come in contact with the anther and the stigma. The body of animal get a coating of pollen grain, uh, coated of pollen grain, which are generally sticky in animals' pollinated flower. When the animal carry pollen on its body come in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe place to lay egg. An example is that of the latest flower of Amorphophallus. The flower left itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant yucca, where both species, moth and the yucca plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of ovary and the flower, in turn gets pollinated by moth. The larvae of moth come out of the egg as the seed start developing. Why don't you observe some flower of the flowering plants or any other available to you: cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animals visit them and whether they could be pollinators. You'll have to patiently observe the flower over a few days and at the different types of days. You could also try to see where there is any correlation in the characteristics of a flower to the animals that visit them. Carefully observed if any of the visitor come in contact with the anther and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as pollen nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your effort. Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination result in breeding depression. Flowering plants have developing many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptive are not synchronized. Either the pollen is released before the stigma become receptive, the stigma become a receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. Third device to prevent inbreeding is self-incapability. This is a genetic mechanism and prevent self-pollen from the same flower or other flower of the same plant from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in pistil. Another device to prevent self pollination Another device to prevent self pollination. अब कहाँ गया बे? Another device to prevent self pollination is the production of unisexual flower. If both male and female flower are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not genetogamy. In several species such as papaya, male and female flower are present on different plants. Thus, is each plant is either male or female. Dioishi. This condition prevents both autogamy and genetogamy. Pollen pistil interaction. Pollination due Pollination does not guarantee the transfer of the right type of pollen. Compatibility pollen of the same species as the stigma. Often, pollen of the wrong type, either from other species or from the same plant, if it is self incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is one of the right type compatibly or of the wrong type incompatibility. If it is of the right type, the pistil accept the pollen and promote post fertilization event that lead to fertilization. If the pollen is wrong type, the pistil reject the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and the pistil. The dialogue is mediated by chemical combination of the pollen interaction with those of pistil. It is only in recent years the botanists have been able to identify some of the pollen and pistil interaction, pollen and pistil component, and the interaction leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatibility pollination, the pollen grain germinates onto the stigma to produce a pollen tube. Through one of the germ pore, the content of pollen grain move into the pollen move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reach to the ovary. You would recall that You will recall that in some plant, pollen grains are said in at two-celled condition, a vegetative cell and a generative cell. In such plant, the generative cell divide and form the two male gamete during the growth of pollen tube in the stigma. In plant which shed pollen into the three-celled stage condition, pollen tube carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enter the ovary through the micropyle and then enter one of the synergy through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropyle part of the synergies guide the entry of pollen tube. All these events from pollen deposit on the stigma until pollen tube enter the ovule are together referred as a pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition, followed by promotion or inhibition of the pollen. The knowledge gained in this area would have the plant breeder in manipulating pollen pistil interaction even in compatible pollination to get desired hybrid. You can easily study pollen germination by dusting some pollen from such pollen, uh, dusting some pollen from flower such as pea, chickpea, Crotalaria, balsam and vinca on a glass slide containing a drop of sugar solution, about 10%. After about 15 to 30 minutes, observe this slide under the low lens of microscope. You are likely to see pollen tube coming out of pollen grains. A breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior variety. Artificial hybridization is one of the major approach of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used to form pollination, and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. In the female part, bears bisexual flower, removal of anther from the flower bud before the anther dies using a pair of forceps is necessary. This step is referred to as emasculation. Emasculation flower have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attained receptivity, mature pollen grains collected from anther of the male parents are dusted into the stigma, and the flower are rebagged and the fruit allowed to develop. If the female parents produce unisexual flower, there is no need of emasculation. The flower, the female flower bud are bagged before the flower open. When the stigma became receptive, pollination is carried out using the desired pollen and the flower bagged. चलो जी, ये पढ़ेंगे पार्ट टू में। पार्ट टू में मिलते हैं।